Hello everybody, I am Shrobana Mukherjee and welcome to my channel Scale Up Science. So today's topic is the amino acids. Okay, so what are amino acids exactly? Amino acids are the monomers of proteins. The monomers are the single subunits that forms something by joining together. Okay, so suppose this is an amino acid and this joined together and this made what? Protein. And this is the polymer. Okay, so you understand that amino acids polymerize to form protein, right? You can take any protein in the world, any, okay, ranging from the hemoglobin in our blood, okay, the luciferin that exists in the fireflies due to which they light up in the dark. You can take keratin protein, like that is in our hair, that is in rhino's horn. You can take any protein in the world, okay, that will be comprised of or composed of the 20 amino acid that exist in the biological world. Okay, these all of the proteins, they are made up of that 20 amino acid. They are combination and sequencing of those specific amino acids. Okay, so why to study amino acid? Okay, so amino acid is very important to study because in enzymatic functions, in later in biochemistry, in any chapter you will study, this will be the foundation topic for you. Okay, before I further go to the functions of amino acid or anything deeper into amino acid, you need to understand what is chirality. Chirality. So, what is this thing? What is chirality? Well, chirality is, suppose now I have two hands, right? I have two hands. Suppose I have four hands, okay? From here also I have two hands. So I have four hands now. In this hand, I have an orange. In this hand, I have an apple. In these two, here I have a banana and here I have say jackfruit. Okay, so in my four hands I am holding four different fruits. So I am a chiral human. Okay, like that. What is a chiral molecule? Let me take the example of a carbon molecule. This is a carbon atom. Okay, so here this is a chiral molecule, right? Why? Because it has four different groups present in each hand of the carbon. Okay, that is called, this is a chiral molecule or this is a chiral carbon. Okay, and my friend, this is also the structure of the amino acid. Okay, only the R varies. The R varies from different amino acids, but everything else remains the same. Okay, you should always keep in mind, if there is a chiral molecule, there must be stereoisomerism and there must be optical activity. These are my two hands, right? This is my left hand and this is my right hand. Okay, so these are my two hands. Now let me superimpose this together. Okay, are they matching? No, they aren't. So these are non-superimposable mirror images of each other. Means if I take this hand and I stand in front of a mirror or I place a mirror here, it will exactly look similar to this. So these two are non-superimposable mirror images of each other. Why non-superimposable? You cannot superimpose each other. Okay, it won't match, right? In biological system, there are many more examples of this type of stereoisomers. Okay, so this is called as enantiomers. The non-superimposable mirror images are called as enantiomers. The example of enantiomers also includes our ears. These two ears are non-superimposable mirror images of each other. Our foots are also non-superimposable mirror images of each other. This is also a kind of stereoisomerism, and that is called as enantiomerism. These are called enantiomers. Okay, so this is my right hand, sorry, and these two are enantiomers. Let us look at the example of one amino acid. Okay, that is called as alanine. What? Alanine. Alanine. Right. Sorry. Hope you can see it. Okay. Here. See, where is the NH3 molecule? It is on right hand side of carbon in respect to carbon. It is on right hand side. Okay. So where is the NH3 molecule? It is in right. So this will be called as the D enantiomer. And C, where is the NH2 molecule? It is in left. So it will be called as the L enantiomer. Okay, L enantiomer. You understand that? When in right side, always take for the cases of amino acid, always take the NH3 molecule. If it is in the right side, it is D enantiomer, left side L enantiomer. Okay. Now, so this is D alanine and this is L alanine. Right? What is optical activity? Remember, I told you, for any chiral molecules always show enantiomerism. Not enantiomerism, I will call it stereoisomerism. And these are optically active too. Optically active. Now you know about stereoisomerism. So next I will talk about optical activity. What is optical activity? Okay, so suppose this is a sodium lamp, okay, and it has light coming from many directions, okay. This is a what? Sodium lamp. Now I use a filter to filter this light and make it the plain polarized light. So now this is the plain polarized light. Plane polarized light. Okay. Now I am passing the plane polarized light through a polarimeter. What is a polarimeter? It's an instrument. Probably we'll see it in the labs of your colleges or schools. You have seen it. There is a tube like this. Okay. So, okay. And now here I have the solution of the compound that I will check the optical activity. Okay. I will be checking the optical activity of this solution. Right. This is the solution of that compound. Okay. Now it is passed through this, and then here is an analyzer through which I will see and record the results. Okay. So this is what? This is an analyzer. Okay. So the light came out, and you can see it is not in its previous position anymore. Okay. This is my eye. I am seeing here from here, and I am recording that the analyzer was in vertical position, but the observed rotation means I have to rotate the analyzer in a clockwise direction to see see the light, plane polarized light. Okay. So the change in observed rotation in this case is clockwise. Why clockwise? It was in vertical position, the analyzer was in vertical position, the plane polarized light came, and I needed to rotate it in a clockwise direction to actually see it. This is called the positive observed rotation, and this will be called as dextro rotatory or plus D, you can tell. Okay. If the light would come like this, and it is the analyzer, vertically it is this is my eye. Okay. Now I have to rotate it like this. Now what is the observed rotation? It is anti-clockwise. Anti-clockwise means minus, and this is called levorotatory. That is minus L. Okay. For dextrorotatory, it would be plus D. So plus D and minus L. Okay, do you remember L-alanine? Let me draw the structure first. This L-alanine has both minus L and plus D forms. Means it rotates the plane polarized light in anti-clockwise direction and in clockwise direction as well. How many amino acids are there? 20 amino acids are there. Among 20, glycine is a chiral amino acid. Okay, means in CH3 instead of CH3, glycine has H. So it's not anymore chiral, right? There are no 4 different groups at all. Glycine is an exception among the amino acids because it has H. Okay, so among 19 amino acids that shows stereoisomerism and optical activity, there are only 9 among the 19 L isomers of L enantiomers of amino acids, only 9 of them shows only 9 of them are dextrorotatory and rest 10 are levorotatory. Okay, 9 of them are dextrorotatory and 10 are levorotatory. Okay, but this form, levo form, this is very much abundant in biological systems. It takes parts in many enzymatic reactions and many other biological functions are carried out by the mostly levo amino acids. Okay, so you know why you always see the drugs in levo form, like for example the levocetrizine, right, levosalbutamol, these are the examples, that's why. Okay, it seems very easy here to see that there must be a levo form, there is a dextro form, there is a dextro form, there is a levo form, right, in optical activity. But when you synthesize it chemically in a lab, when you synthesize any amino acid or any molecule that has a chiral center in your lab, it is very difficult to isolate the dextro form and levo form. It is very difficult. Why? Because they will come in racemic mixture form, means these two combinations. You will see the two combinations, the mixture of that compound. So how will you isolate the levo form from the dextro form? So in lab, how will you differentiate levo from dextro? There are very few processes. First one is by enzymatic reactions. You can use any enzymes where specifically, which is the levo form will only attach with that specific enzyme and the dextro will be precipitated. Okay, so you will get in the bounded form with the enzymes the levo form, you will get that. Number 2, recrystallization. By recrystallization also this thing can be achieved. Number 3, chiral column. You can use chiral column for chromatography, like in GC or in LCMS, chiral columns can be used. Okay, or you can also use XRT, means X-ray diffraction techniques, for the isolation of this. Okay, so this is all for today. I have discussed about the stereoisomerism, the optical activity of amino acid and some basic things. Okay, in next chapter I will be talking about the structures of amino acid and their chemical activities. Okay, so I hope you have found my video interactive and insightful. So thank you so much. If the video has been informative to you, please subscribe to my channel and book bell like button.